Okay, so first of all, I thought I'd give you a single slide about Sweden and Europe, because Sweden is actually one of the biggest countries in Europe. We're at number 5 with 450,000 square kilometers, compared to, say, Germany at 357, Italy and United Kingdom. So we're roughly twice the size of the United Kingdom, if you don't count the overseas territories. On the other hand, Germany is number 2 in population at 82 million, and United Kingdom and Italy are about 60 million people at 5 or 6, and we are at number 17 with 10 million people. So we are a fairly big country with very little people. That has, of course, led, especially during the 19th and 20th century, to quite a bit of regionalization. So there is quite a bit of autonomy in separate regions, and Sweden now consists of 21 different regions, 7 of which have university hospitals. So this is Region Västerbotten, Region Uppsala, Region Stockholm, Östergötland, Örebro, Västra Götaland, och Skåne. So all of these have a university hospital, and then we have 14 others that don't. Problem is, of course, that healthcare budget is set by the region because it's regionally controlled, which means that you have to contend with 21 different political bodies for funding, and each region will have its own priorities and interests linked to the certain politician that seems to be the one that sets the budget. There's also quite a few issues between sharing data between these regions because all of them are separate legal entities. So healthcare data there are ways of sharing data, but it's kind of depends on which lawyer you talk to, how easy and how you can do that. It also has also led to heterogeneous diagnostic standards because depending on the budget you have in different regions, they will have prioritized different technologies as well. So if you go to Västra Götaland, for example, with cancer, you might get some different diagnostics run on you compared to, say, Region Kalmar or some other small region. We also have a plethora of EHR systems, so it's like a jungle. I think it's 1600, 1700, something like that. I know that at just my single hospital we have like 16, and these have all grown during the last 30 years. So some are new, some are not. Some were designed to cooperate and interoperate, some don't. And it's quite a bit of a mess. And for the last 20 years or so, we have tried to rectify that, but it's an uphill struggle, especially since if you want a national EHR system, all of the 21 regions have to agree, and all the 21 regions have to put in money, which is difficult to achieve. Each region also has a very small patient population. Västra Götaland is one of the biggest with over 1 million people, but some of them are maybe just a few 100,000 in a single region. And this is also a little collaboration, of course, because there are so many legal and budgetary issues that sort of prevent collaboration. So how do we actually perceive precision health? I changed this from precision medicine because everyone else is talking about precision health now. Precision medicine is so 2018, apparently. So how do we achieve this in Sweden? Well, what we did was we created a project called Genomic Medicine Sweden. And Genomic Medicine Sweden is actually a collaboration between 7 universities and 7 university hospitals. So it's all 7 university hospital regions in Sweden, as well as all the 7 universities. So this is not a separate entity compared to healthcare in general. This is something that's grown from inside healthcare and out. And this is done through regional collaboration between these 7 university hospitals. So that differs as compared to, say, Genomic England or Genomics Ireland, where you have a separate entity that's sort of spun off from the National Health that does this kind of precision medicine thing. This is actually from inside healthcare and moving outwards. So our mission statement is to introduce modern diagnostics using NGS, and to do that with sort of implement precision medicine. And by precision medicine, we mean the correct treatment for everyone. So improving diagnostic methods and making sure that the right people get the right drugs. We also want to foster national collaboration, improve that, which isn't there, hasn't been there traditionally. Harmonize care across regions, so regardless of where you go in Sweden, you will have the same type of diagnostics run on you if you have the same indications. And with that, we can also create very unique national research opportunities because we're already pretty good at keeping track of our citizens. And we have quite a few quality registries. We have quite a few national registries and biobanks, stuff like that. And we are in the position now that we can start linking all these groups together. And we also want to further industry innovation and collaboration, especially with both diagnostic companies and the pharma companies. And to do this, we're creating genomic medicine centers. So that is a new national structure and infrastructure in Swedish healthcare. So we're actually reshaping part of the diagnostic labs in all these 7 university hospitals to create a genomic medicine center that can collaborate with other genomic medicine centers. We started this in 2017 with a pre-study. This is in Swedish SEK, but if you divide this by 10, you will get approximate euros. I don't know what the pound is at the moment, but well. So we started in 2017. 2018, we got another 80 million Swedish crowns, which would be 8 million euros. That will last us up to December 2020. And then we have a continuation phase. We're right here at the moment, where we're going to need about 150 million euros to implement this in Swedish healthcare. And then we're going to do that for approximately 10 years, where during that phase, we are going to phase ourselves out, and all that we do is going to be moved into routine healthcare. So this is not something that's, this is an implementation project in the sense that we're not going to do 200,000 genomes and then call it quits. This is something that we're trying to move into diagnostics and healthcare. This is the old braid. I have a new braid, but this is our organization, and we have technical packages on the horizontal axis and medical packages on the vertical axis. So we are focusing on rare disease, hematology, solid tumor, complex disease, and microbiology. Where we have national reference group for example, the solid tumor group is 160 pathologists from all the 7 universities and university hospitals. Rare disease is essentially every clinical geneticist in Sweden. Same thing with hematology and microbiology, and we just started complex diseases where we work with GAPS, the Genome Aggregation Project in Sweden. So this is also a bottoms-up approach where we actually ask the clinicians what do they want, what do they need, and what would they like in the future, and then we try to make that happen with their effort and with their help. On the technical side, we're looking at sort of the legal and ethical frameworks, because right now they're a mess, and we need to change a couple of laws for this to be possible in the future. Biobanking, sequence data generation, analysis and interpretation tools, data storage, data organization and sharing, as well as education and health economics. And I'm actually involved in most of these groups. But my main focus is data organization, sharing, and interpretation analysis. So now we have 7 genomic medicine centers. I am coordinator for GMC West in Västra Götaland. And I have 6 other people that work with me in the different regions to sort of bring this into healthcare in the individual region. So it's all collaboration here too. Each GMC is organized by the host university and university hospital, so it's actually a shared organization where we can bring in resources both from the university and from the translational researchers at the universities, as well as the actual clinicians and pathologists and clinical geneticists that work within routine healthcare. They in turn represent each region, so each GMC represents its region in the national effort, but they all support specialized care in their region, and they have this wide range of molecular diagnostic tools. So the point isn't that we're going to move all cancers to Stockholm and all rare disease to Skåne. The point is that we need to have harmonized care where each region can perform as many and as advanced as analyses are needed in that region. And all of this is, of course, part of the national coordinated effort. So we have national reference groups that sort of decide and agree on what needs to be done in healthcare, and then it's the GMC's mission to actually implement that in each region. So when it comes to data generation and data storage, we're creating something called the National Genomics Platform, which is a data lake where all these 7 university hospitals will input their raw data, QC data, diagnostic data, and variant data to start. All that goes into the lake. In the future, we'll also add treatment data and economic data. We'll also link this up to the biobanks and the quality registers that we have in Sweden. It's like 16, 17 different ones, as well as e-health records where it's possible. So there are a few systems that we can link up. We have Cerner Millennium, we have Cambio, and we have Epic, different HR systems that we're going to link into this. Mostly to create interfaces so you can pull data into EHRs from the National Genomics Platform, and as well as pull some stuff back from EHR records into the data lake. Because on the other side, we can get epidemiology, decision support, and statistics out of this. And in the future, we can also get, with treatment data and economic data, we can get efficacy and health economics and research data. And I should add a final point there that I didn't bring into this slide, but I have in my next version, and that's AI. So one of the big missions in Sweden right now is how we can apply AI in healthcare. So we have AI Innovation of Sweden, we have the Wallenberg Center for Quantum Technology, a couple of different large research initiatives that all focus on how you can apply AI in healthcare in various forms, from logistics and how you plan beds, and how you plan various tasks in healthcare, to how you interpret data, how you interpret variant data, and how you link that to phenotypes and genotypes. This is a more detailed view of what I'm going to do, or what we're going to do. So we're actually going to use cloud computing to do analysis, because it's incredibly cheap and it's available in Sweden. So Amazon is actually, so the host of the Nordic region in Amazon is Sweden. So we have 3 data centers from Amazon in Västerås, Eskilstuna, and I forget the third one, but it's in the middle of Sweden. We will use or create a tool called NGP Commander to do analysis and data browsing, which will be individual for each region. So each region will have its own little commander that can do all this kind of stuff. But it will be linked up to the NGP repository, where we'll store all the data. And in the repository, we will have 2 layers. So you will have the private layer, which is the black ones down there, where each university hospital can input all kinds of data without actually sharing it. Then you will have a layer between, where you can decide what and who to share with, and sort of access control list, stuff like that. And then you will have a public layer where you can share certain types of data with all participating regions. On top of that, we have something called the NGP Intelligence, where we do population genomics databases. So we can actually aggregate in real time all the data comes into the repository up to the genomic database. So you can have a real-time view of exactly what kind of variants, for example, who has classified what into what, and so on and so forth. And that all goes into the decision support platform. The other platform is for advanced visualization, and that's a request from epidemiologists and microbiologists, because they want to track national infections. So we're doing that together with the Public Health Agency in Sweden, where we want to have a single platform where both hospitals and the Public Health Agency can track national infections in real time. So this is my little piece of the world, GMC West and Sahlgrenska University Hospital. It's located in Gothenburg, and it was founded in 1772 by Nicholas Sahlgren, therefore Sahlgrenska. When we started, it was 24 beds, 14 for paying customers, so 10 beds for free and 14 for paying. And Per Dubb was the first Admiralty Medicus, and I think that's the most wonderful title I've ever seen. If I ever sort of get to pick my own title, it should be Admiralty Medicus. Now we're at 2,300 beds, and we're about 17,000 employees. And we serve 1.7 million people. We're doing quite a bit of NGS already. So right now we have a couple of MySeqs, NextSeq, PGMs that we're sort of killing off one by one. The only one we have left we're using for virology research. A couple of S5 primes, and a NovaSeq that we use for everything else. So all of these others we're sort of slowly phasing out in favor of one other NovaSeq, so we can have 2. When I started at Sahlgrenska in 2016, we were divided into clinical genetics, hematology, and pathology by tradition, and we were running a bunch of different panels. I think actually for clinical genetics there were a couple of more ones, and I think maybe it's cut off there too. And everyone was doing these panels, everyone was doing the local data analysis. So when you would have data coming from the instruments, you would load that into your computer and you would run some sort of application, and then you would upload that to either Cartagena or IVA or Illumina Variant Analysis or iReporter, depending on where you were at the time. This is, of course, incredibly inefficient, especially since pathology, they picked their own instruments, so they were running like their whole pipeline by themselves. So when I started, I thought that this is not what we want. So we started a concerted effort to move away from this. So by 2018, first of all, we had started trying panels. So most of the whole exome, the panels were done with whole exome sequencing for clinical genetics. We still had 2 panels for hematology and a couple of different ones for pathology, but now all the data analysis was done on a single cluster that I set up with 500 cores or so and a couple of 100 terabytes of disk. So we started sort of collecting all the data, at least, that came out of the instruments into and doing sort of a concerted and organized analysis. We still use 3 different decision support tools, but at least we're sort of moving in the right direction. So by 2019, we moved directly to whole genome sequencing. So everything that comes in that is rare disease in the VG region today would be whole genome sequence. Then we have in silico panels to pick out the interesting parts, but everything is done in the same flow and everything is whole genome sequencing. For hematology, we're actually using a single GMS panel as well as whole genome sequencing for leukemias, where the GMS panel is the same panel as run at all the other 7 university hospitals. So that's a single panel for all hematology with about 250 genes. Same thing with pathology, where we're moving to a single GMS panel for all solid tumors with about 550 genes, I think, where everyone will be using the same panel, which means it's instantly comparable between regions. And we can all collect this in a single place, which is great. We're also using ultrasensitive sequencing for minimal residual disease during treatment. That's a method that's actually developed by a group in Gothenburg, where we can detect very extremely low amounts of cell-free DNA. So we can use that during treatment to look for emergence of resistance mutations during treatment. And finally, we do pediatric cancer whole genome sequencing. We've been done that for a while, but now we're actually moving from just doing a pilot with a few different diagnoses to all. So all pediatric cancers in Sweden actually will be whole genome sequenced. We're also slowly moving away from our nice little cluster, and we're actually doing cloud bursting now. So we're starting to cloud burst analysis up to Amazon to reduce costs and improve efficiency, actually. And we're also selling on 2 different systems for decision supports. It's either Cartagena together with Moon mostly, and QCI for hematology and pathology. Finally, I mentioned this at the earlier meeting, but I thought I'd make a few slides about it. So this is a case that we had with a boy with neuroblastoma. So neuroblastoma is a tumor in the sympathetic nervous system in childhood. So you get all these tumors, and it spreads very quickly. It's almost always metastatic. And in neuroblastoma, you have biology that directs treatment. So depending on the biology of the tumor, you will use different types of drugs and different types of indications. As you see up right here. So we had this little boy. He was 16 months when he came in, and he was metastatic with neuroblastoma, and he had an 11q deletion that you can see up here. This is his array profile, and you can see that you have the 11q loss here. And if you paid attention on the last slide, you can see that, of course, if you have 11q, you use DSB inducers. So double-strand break inducers, but there is a rather harsh treatment that introduces double-strand breaks all across the cells, with the idea that the neuroblastoma cells are slightly less capable of repairing that, so they will die more than anything else. Now, this little boy got extremely sick from this treatment, so he was referred to the ICU. He had daily transfusions for almost 6 months because he got extremely anemic. So he was very, very sick. And during that time, we couldn't really figure out what was wrong with him, so we did a whole genome sequence of the tumor and the normal. And we found that he actually had a very rare, undiagnosed rare disease called funky mutations. And funky mutations, what they do is that they prevent the cell from repairing double-strand breaks efficiently. So essentially what we're doing when we introduce double-strand break inducers is that we destroy an equal amount of healthy cells as well as neuroblastomic cells. So that explained why he got really sick. But we also find an ALK mutation, which is actually targetable. So we can actually move him from the DSB inducers to ceritinib, which is an ALK inhibitor, second generation from Novartis. And it worked wonders. He got better. It was actually back in preschool. And this is him, actually. So this is Wilfred, is his name. So he's now 6 years old, and he survived thanks to whole genome sequencing and precision medicine. This has sort of been a banner case for GMS as well. So this is one of the cases we like to bring up because this is one instance where this actually worked. It didn't work with traditional methods. It didn't work with array. But when we did a whole genome sequence, we can actually find both what caused the reaction to the medicine and also introduce a better medicine and give him sort of back his life. So this is him and his sister. So he's 6 now. And with that, I'd like to finish. And of course, if you want to know more, you can look at genomicmedicine.se. The English site is a little underdeveloped because we focused on the Swedish site. So I'd like to apologize for that in advance. So thank you.